Hi, my name is Julie Thompson, and in this video, we're going to talk about DNA mutations and repair. Before we start our discussion of DNA mutation and what repairs these mutations, I want to think about what causes them in the first place, and I'm going to provide a few examples. All of us produce reactive oxygen species as a result of metabolism, and these reactive oxygen species can damage your DNA. That's not something we can control our exposure to, but the carcinogens in cigarette smoke you certainly can, and those are known to cause DNA mutation. Exposure to X-rays or UV light can also induce damage. And then finally, DNA polymerase, it's the enzyme that copies our DNA. It's not perfect. It can make mistakes, and that can lead to mutation as well. Here are the different mutations that we'll explore, along with their corresponding repair mechanisms. Sometimes the damage is pretty localized, so damage to a single nucleotide or a small number of bases. Sometimes there's just mistakes in replication, so it's not damage per se, but the wrong nucleotide is inserted. And then sometimes the damage is more extensive. So for example, the DNA can undergo what's called a double-stranded break. Okay, so let's first consider the case where a single base is damaged. In this case, your cells have a mechanism known as base excision repair. The DNA repair enzymes will remove that base. DNA polymerase will insert a new base. After the new base is inserted, there's still a nick in the backbone of the DNA molecule, and that's the job of DNA ligase, which will seal that nick. So to review the steps in base excision repair, first there's DNA repair enzymes that recognize and remove the damaged base. Then DNA polymerase will add a new nucleotide. Finally, ligase seals the remaining nick. Next, we'll look at damage to a small number of nucleotides. A common example occurs when two adjacent thymines join together to form a thymine dimer. This often happens in response to UV light. When there's damage to a small number of bases, your cells have a mechanism known as nucleotide excision repair. In this case, DNA repair enzymes recognize the damaged region of DNA and cut out a small area that contains the damage. DNA polymerase will fill in. But the DNA polymerase leaves behind a nick in the backbone of the molecule, so ligase is needed to seal that nick. So let's review. Nucleotide excision repair occurs when there is damage to a small number of bases, and the DNA repair enzymes recognize those bases and remove a small section of DNA surrounding the damage. DNA polymerase will fill in that gap, and then finally, ligase seals the nick that remains. So in the last two examples, there was actually damage to the DNA, but a lot of times mutations result from mistakes in replication. So DNA polymerase, as it copies the DNA, it does a pretty good job, but occasionally it will insert the wrong base. So here we have T pairing with C, which isn't right. DNA polymerase can backtrack, remove the C, and insert the correct nucleotide and continue on its way. So proofreading, DNA polymerase can remove the incorrect base during replication, and it can insert the correct base. It's almost like having a delete key on your computer, where if you type the wrong letter, you can just do a quick backspace and then keep going. Now let's consider mistakes made during replication that are not fixed by proofreading. So in this case, DNA polymerase is copying the DNA, and it inserts a C instead of an A, but this time it doesn't go back to fix it. It leaves the error in place. Mismatch repair functions to cut out the mismatched base along with several bases surrounding it. DNA polymerase will fill in the gap, and it once again leaves behind a nick that is sealed by DNA ligase. So mismatch repair recognizes mistakes made during replication, and the DNA repair enzymes are responsible for removing the mistake along with several surrounding bases. DNA polymerase will insert the correct bases, and then ligase seals the remaining nick. Mismatch repair is very similar to nucleotide excision repair. The difference is that nucleotide excision repair cuts out damaged DNA, while mismatch repair removes errors made during DNA replication. Now let's consider extensive damage to the DNA. So things like X-rays can cause double-stranded breaks to form in the DNA molecule. When this happens, the DNA is not very stable, so some of the DNA may get eaten away. Recall that you have two sets of genetic information. You have two copies of all of your chromosomes. So if the DNA on the top, it represents a region on chromosome 1, you have a second chromosome 1 that has similar genetic information. What this means is that the similar genetic information, or we say homologous, can be used to repair the broken DNA. So in this form of repair, which is known as homologous recombination repair, the information from the intact second copy is used to repair the damage. So in homologous recombination repair, double-stranded breaks can be aligned with the homologous second copy of the DNA. The undamaged DNA provides the needed sequence to repair the damage. Double-stranded breaks that form due to something like X-rays can also be repaired by non-homologous end joining. So we have our double-stranded break. DNA is not very stable when it has these breaks, so you can see we lost some of the nucleotides. Rather than using the second copy of the genetic information, in this case, the cell simply glues the DNA back together. And you can see that some of the genetic information was lost, so this is really a last resort mechanism. So non-homologous end joining, it repairs double-stranded breaks by taking the broken ends of the DNA and gluing them together without regard for sequence. So some genetic information can be lost. I hope this video has been helpful. Thanks so much for watching.